From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 71, recorded on September 21st, 2021. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey there, Vincent. Great to be with you from kind of modified LD Lab Studios. I'm in a temporary location for a few minutes. Speaking of which, it sounds like you've got a location upgrade in the works here.
Oh, yeah. As everyone knows, maybe I've been making the incubator.
Yeah, this is exciting.
It's a new recording space in New York City, and we are almost ready to record. In fact, I'd wanted to do this from there, but actually the timing, I had to come up to Columbia here and do some mouse work with Amy. Mm-hmm. So I couldn't, but I think Thursday evening's episode with Daniel Griffin will be the first of many to come from the incubator.
That's fantastic. Yeah, I recognize the backdrop there where you are currently. I've been there on a couple of occasions, but this just sounds like a really exciting new chapter.
Yeah, I mean, this is, my office where I've been since, 38 years. And, you accumulate a lot of stuff. So it's going to take a while to get to that point. Right now, the incubator has white walls.
Yeah. Yep. Work in progress. I'm guessing, knowing a little bit of your science style, that it won't be long before there are incredible images of virus structures and various other things that come into play there.
I have big ideas for the wall behind me. Right now it's a white wall, but yeah, we'll put stuff up. But I'm not going to have a couch with a phage pillow or pictures, but I'll find some place for those in the incubator.
Yeah, sure. That'll be fun. Can't wait to see what happens in the microbe.tv of the future here.
Indeed.
Well, Vincent, we're back from summer break. We've been long time
I don't think you took a break. I don't think you took a break.
Well, I had a really fun conversation with author and science communicator Carl Zimmer last time.
Yeah, that was great. I enjoyed that.
Glad. And thank you for your trusting me, turning over the keys here of TWiEVO while you were called away, I think, to NIH to do some consulting, it sounded like. And so we just decided to go ahead with it.
That's correct. Which didn't take very long, unfortunately. So
gotcha. Yeah. But no, and then we're a little, I think August got a little bit busy, and so it's good to take breaks from now and then. But we're back, and I think we have some really fun stuff kind of in the hopper this autumn and this fall. A couple of guests on the horizon. Stay tuned on that front, and then of course there's plenty more to talk about. It's been a couple months, and there's been a lot going on coronavirus evolution wise, and so I guess that's what we're here to do today.
So, by the way, you're missed. Look at that.
Thanks so much, Priscilla. It's great to be back. Great to be back live streaming, actually. So we did, the last one was a recording, and I have to say, Vincent, there's something about the adrenaline flow of live streaming. Can see the eyeballs coming up in numbers here slowly and surely. It's just really great to be part of this bigger community, and good to be back with all of our listeners. It's great to see you a kind of a moment's notice back in the game with us. Really fun.
Yeah, I'm trying to move my name here, and now I made it all too big because we changed the aspect ratio of our images just before coming
that's
on.
Right.
And I guess you can see the chat. And there's another one here. Let's see. This is pretty funny. Remember, this is YouTube. No productive or positive comments.
Point well taken, Michael. We'll try to take it down a notch or two on the optimism and fun.
And there's hi from Sweden. And Vincent was recently called out as a fraud with his definition of virus isolation in a viral video. Yeah, I'm a fraud. 40 years of virology, and I'm a fraud. Well...
Uh-oh.
Oh well. That's life on the internets right now.
That's right. Well, and we'll kind of do a little visit on some controversial topics. What is the Delta variant versus the Alpha, competition between it? There's some really fun things for us to tackle here. So before we get to that, Vincent, you pulled up this really interesting paper on mutation rates. How did you get onto this one?
Let me pull it up here briefly so people can see it, since we have the wonders of the internet. This is now, I believe, in press at Genome Biology and Evolution. Because if you go to the website, it looks like it's in press, right?
Yep, that's right.
This was sent by a listener, a TWiEVO listener, and I had meant to read his email, but I can't find it.
Oh, no.
No sweat. Not surprising, because I just have too many emails. But this is causes and consequences of purifying selection on SARS-CoV-2. And when I saw it, I thought this is right up Nels's alley. This is all about, purifying selection and mutation rates. So, in fact, I need you to understand it to a certain extent.
Well, we'll do what we can. But this is, I'd say it's right up our alley. It kind of actually harkens back in a way to TWiEVO episode number 2, where we talked about, this sort of simple property, the mutation rate. What seemingly seems simple is actually a lot more complicated when you start to dig in beneath the surface. So we looked at a paper, I think it was published in eLife, on the mutation rate in HIV, and some evolutionary analysis suggesting that the mutation rate is sort of underestimated. We actually, I don't know if you remember, getting to Michael's point about not being too optimistic, or, this is on YouTube. We did get some complaints about our our second episode. It felt like we were maybe stumbling out of the blocks. One of the sort of giants in HIV biology and evolution, John Coffin, thought we were kind of taken for a ride on that paper. But I think it actually, in retrospect, kind of highlights how still argued over the mutation rate is. These sort of what you'd think is like a fundamental just property that you could measure pretty easily, and it's used, of course, in all kinds of calculations to think about how to try to predict a virus as it changes, or to look backwards to think about where did a virus come from, when was the last common ancestor. You need mutation rates to kind of plug into these models in order to make these estimates, to get a kind of quantitative sense. So a really important fundamental thing, which turns out to be a lot more complicated when you start to dig under the surface. So let's set this one up. This is as you pointed out, just published in GBE, Genome Biology and Evolution. The first author, Atahualpa Castillo Morales, and last author, Lawrence Hurst. This is all coming from the Milner Center for Evolution, University of Bath in the UK. Seeing some collaborators here, also at MRC, Human Genetics, and then at Aarhus University in Denmark.
Do you know any of these people, Nels?
Yeah, so by reputation, Lawrence Hurst, sort of, a long-time practitioner of evolutionary biology. I think trained even with Bill Hamilton, sort of this great character, and one of the sort of founders of the Red Queen hypothesis, which certainly colors or influences a lot of my own work. And so and they were, I think coming at this question, what is the mutation rate of SARS-2? And so, even from first principles, you'd almost think we'd already have a pretty good handle on this. We'd already know what this is, and easier said than done. So I think the authors rightly kind of set up the kind of state of the field, and note that actually I think it's a great paper to just think about, to step back and just think about in kind of simplified terms what's really going on here with mutations, and the fact that it's so the first step of a mutational process, whether it's a virus, whether it's a host, whatever living creature it is, the mutations happen, and then selection acts on them. And those are two different things: the random process of mutation, and then the more what looks like the directed outcome of natural selection. And so, the authors here, as you pointed out, Vincent, are thinking about purifying selection, the removal of mutations from a population because they're deleterious. And that's kind of the setup here, is that they're proposing that, if you just go, and the resources now are vast. I think we've talked about this on GISAID, this database where people are depositing SARS-2 genomes. I think I just heard in the last week or so that, the US was a little bit slow to get into the game here in terms
Yeah.
of the genome surveillance of SARS-2. That started to pick up. I think we just passed 1 million sequences that have been deposited either in GIS8 or kind of some related databases. And this is kind of dwarfed by the world's contribution to this genome data set. Many more millions of sequences now available. And so people, as the authors pointed out, have started to calculate a mutation rate or tried to start to analyze like can even in this sort of real time, coming up on 2 years of sampling, natural experiment, horrible pandemic related experiment, or other signs of selection, what's the mutation rate? And so I think kind of the almost even conventional knowledge in the field is that if you calculate this ratio, the so-called Ka to Ks ratio, also kind of another way of, or maybe more common way of expressing that, the Dn to Ds ratio. This is the rate of non-synonymous substitutions to the nucleic acids divide by the rate of synonymous or silent changes. And as we've talked about many times on TWiEVO, we have the degeneracy of the genetic code, where if you make DNA or RNA, in the case of an RNA virus, letter change, whether that codon codes for a different amino acid because it's degenerate, not the answer is not always. And in fact, and this will become a kind of central to this analysis in this paper, the so-called fourfold degenerate codons. And these, right? So these are, there's in codons, actually remind us, Vincent, how the 3 positions in the codons signify sort of functionally important things.
The first and the second and the third base, right? The third base
exactly.
is the wobble. The third base is the wobble, which can be many different, one of 4 bases in some cases coding for the same amino acids. And the first and second do not wobble, right?
Yeah, exactly. So it's not, each position is not created equal. And so
right.
If we change that first and second, you're going to change the amino acid, but the third has that flexibility, that wobble, classically defined. And so the fourfold degenerate means whichever one of the 4 bases you choose in that third or wobble position, you still code for the same amino acid.
So they're using this to try and get a handle on the mutation frequency, which they say we're missing. If we use DNDS, we're missing a lot because all the bad changes we never see, right? So we can't measure them.
Yeah, yeah, correct. So what they say is that, if you just take, you dig in or sample some of the, like these genome-wide data sets, and by the way, easier said than done. There's like any of the science we talk about, if you go a little bit deeper, it's more complicated. Are there sequencing errors in there? What's the quality if it's as these are deposited from all different laboratories around the world, or public health outlets or diagnostic labs, et cetera? There can be a pretty big range in the quality of the data that you're analyzing. But if we just assume the sample is decent and good enough, then you can start to calculate these values by aligning and comparing the substitutions or mutations that are accruing. And so I think the estimate they hold up is that Ka, Ks, or DNDS is roughly 1 in some of these comparisons. And that's a little bit of a head scratcher. So, so the sort of the 3 kind of key areas of the ratio or value of the ratio that evolutionary biologists think about is if it's if that ratio is less than 1, being diagnostic of purifying selection, which is what we're talking about. A mutation happens, it's deleterious, it's purified out of the population. You don't see it. It's what they describe as a missing mutation, because it's pulled away from the data sets before you would capture it. Those viruses don't replicate. The genomes don't propagate. A value equal to 1, which was some of the estimates in the previous work that the authors are holding up. This idea is that can be diagnostic that selection's not acting on the mutations. So it's, probably in a more biologically relevant situation, very weak selection. Obviously, changes are happening here, but it's a very minuscule thing. And so that was if you take that at face value, then basically the rate of, or the number of mutations you see is the total that's there. There's none missing because they're not being purified out of the population. And so that was sort of what I think got them onto this analysis. And we'll dive into this a little bit, that fourfold degeneracy. I do have to throw in my favorite TNDS ratio, which is greater than 1, positive selection, this is
Mm-hmm.
where a mutation can actually be beneficial. And so this
Yeah.
is where,
Yeah.
my lab, a number of others, we can't get enough of this, like to identify these cases of recurrent positive selection, especially as it relates to virus host interactions, and then try to chase down, is there a
Yeah.
story here? Does the variation matter for whether I'm going to be, do well, or my species will do well against a specific virus or another infectious agent, or whether the microbe will have the upper hand, kind of a situation.
I think the point they make, which is really important, is that these DNDS are things that happened a long time ago. You need a lot of evolutionary time, and it's not something you can readily discern in a year, right?
That's right. Yeah. Really good point, Vincent. That's exactly right. So those algorithms are kind of tuned and work best for exactly what you're describing, which is fixed mutations
Yeah.
amongst different species. And so there's been a lot of effort and productive effort put into thinking about virus evolution too. There's an entire website kind of devoted to this, to some degree, called datamonkey.org, or this so-called hi-fi analysis, from some scientists, some genome scientists we featured on the show, some of their work on the show over the years. But even then, usually that analysis is sort of best done where it's like between different strains of viruses, or there's still more diversity in the population. Here, in our current pandemic, I think the authors describe this as like real-time evolution. This is like, the changes are coming off the sequencer in the course of weeks or months, and we're trying to discern the signals. Really difficult to do. With this kind of analysis. Alex Jeffries, who's here, is the
Yeah.
guy who sent you the paper. Fantastic, Alex. Thank you
Good.
for the
Good for you.
suggestion.
Thank you,
Yeah.
Alex. I couldn't find it. I'm sure it's in my email box, but I really appreciate that you're here, and I decided to do it here on TWiEVO. I hope you don't mind. Thank you for sending it in. Now,
Yeah,
it's one of the
great.
coolest things they say in this paper. They
Hmm.
say an ideal examination of real-time selection in the wild would require analysis of massive numbers of full genomes of a relatively fast-evolving species sampled continuously in time and place. Oh, that's exactly what's happening now.
That's right. No, and I agree with this. Like SARS too. I mean, it's unprecedented. Just that the timing of this, where the
Yep.
availability, almost the democracy of genome sequencing, is allowing like sampling far and wide, not evenly distributed, but with some noticeable lags from even some well-resourced places, including the U.S. But we've never like watched a population at this kind of level of
Nope.
resolution, full genome in real time, and then had it like in many cases publicly available. It's really kind of
Yep.
a spectacular thing. So anyway, so the authors, I think, recognize that. They then kind of dig in with this idea that, okay, DNDS or KS might be letting us down. And so they, just as we were discussing a couple minutes ago, Vincent, they kind of want to take advantage of those sort of different properties of the codon position to just ask a really simple question, which is, as we're talking about that wobble site, where in a fourfold degenerate codon, means that selection shouldn't really, all other things being equal, selection probably won't influence or dictate which mutation is there. You could get away with any of the four because you still the protein ends up with the same amino acid at the end of the day. And but then what do you compare that to? So the idea would be that if you look at position 1 and 2, if those positions really do matter, because if you make any change from the original, that will change the amino acid. That's likely then to kind of echo in some cases, at least, into the function of the protein. And so if we look at figure 1, you can see mutations per reconstructed site, and at the third position, for each of the 4 nucleotides, there it is in the blue bars. That number is higher at that site than it is in the first 2. And so that gives us that little kind of hint into the idea that there's some constraint here. There's some purifying selection. If the mutations, there are definitely assumptions here. It's that mutations are happening equally across the genome. It turns out they're not, but that there's still this pattern that's really discernible, and you can kind of take into account some of those differences as well. And so, using analysis, really simple analysis, and then following on with a few kind of complementary approaches, they're able to estimate. I like how they kind of simplify the message here. They say that, for every 10 mutations that you might see in the population. Yeah. So here's another way of kind of slicing this. So now we're looking
like
at
this. Yeah, this is really good.
Yeah, these kind of violin plots, as they're called. So you can see the mutations per site is higher at that fourfold degenerate spot compared to now they're clumping together positions or sorry, no, now they're
by base, right?
Correct. So this is all, it's on base, but it's also twofold degenerate codons, which would be the non-fourfold. And that's actually where now the third position does matter. So you can't just put any of the four and get the same amino acid. If you put two, you'll get one amino acid. If you have the other two of the four bases, you'll have a different amino acid. And so there's some constraint there as well, right? Those kind of for each of the four bases, A, C, G, and U. We're in the RNA world here, so T's or U's. You can see, especially for C's and G's, it's quite clear that the mutations per site are pretty high in the fourfold versus the other non-fourfold degenerate sites. And so very consistent. And so using this, and I think even a couple other sort of complementary approaches, they say that for every 10 mutations that they see, there were probably 5 more that were generated in the population, but selection purified them away. And so that gives you, I think you also included this interesting link that there is like sort of a commentary piece on this. It's sort of like, ripped from the headlines, this thing is mutating 50% faster, which, like, let's pour a little cold water on that. Like, what do we, just the sort of public view or the colloquial view of mutation can sound really scary. Whereas what we're talking about is removing things that are deleterious. Like, this isn't like the virus becoming a superbug. This is like, as you make these mistakes, they're just culled from the population. And
we just didn't see them. We just didn't see them. It's not like it changed suddenly. But what I think is missing is at the very lowest level, the mutation rate is really determined by the RNA polymerase, right?
Yeah, agree.
And the error frequency of the polymerase is the key, which they don't even actually mention, because by the time we get to this analysis, it's quite far removed, right?
Yep,
so
that's right.
The polymerase makes changes. Some of them are corrected because this polymerase does have a correction enzyme associated with it, but some of them get through, and then they end up in virus particles, and then, as you say, some of them are selected against. Some of them are neutral, and maybe a very few are positively selected. So that's where we are. So it's not really the mutation rate. When I think of mutation rate, I think of polymerase error rates, and those are not really determined all that frequently. And, it's hard because any system, let's say an in vitro system using purified polymerase, it would be artificial, and who knows what the relationship to an infected cell would be, right? So it's hard. Yep. But I do think that's the base level that you have to start at, and I'm surprised they don't even consider that in the discussion. Maybe it's too biological,
Well, yeah, but that's why we do this, Vincent. That's why we're podcast, because it's that exact view from virology, from molecular biology, that is kind of missing from the conversation in this very much more kind of population, genetics kind of, or population biology approach. And I think that's vitally important because, and so if you're all working in population biology and not thinking about that, even in vitro mutation rate, then it's all is relative, and of course you want to correct or think about that, but that's another thing that can kind of throw some confusion when we talk between fields, or or then try to express this to the public, like what are we really talking about? And so I agree. I think that's kind of missing here, and your point is really well taken. So in the point that, it's kind of, I think, to a virologist or molecular coronavirologist for sure, and molecular biologist, kind of obvious that there's purifying selection due to the fact that it's very clear that coronaviruses encode a proofreading function. You wouldn't be proofreading, or there wouldn't be selection for proofreading itself if you didn't, if the sort of population wasn't, not to anthropomorphize, but, quote unquote, worried about deleterious mutations. You're actually trying to fix this as a virus, which is pretty unusual.
Yeah.
And then certainly for RNA viruses, very unusual. Probably reflects the massive genome of the coronaviruses. This is one of the super tankers of the RNA virus kingdom. And then the other, kind of to build on your point too, Vincent, the other thing missing here is that, so point mutations are relatively low for RNA viruses, but what's actually pretty prolific is the recombination rate, which isn't, I don't think recombination, it just maybe comes up in passing. And I think that's actually a really important not just a detail about how the virus diversifies, but also matters in some practical ways too. So, depending on how recombination is, like template switching between, in some cases, closely related viruses, there's some evidence we've covered in the last year, pointing out recombination, even probably in co-infection scenarios of SARS-2. But that can also lead to some kind of funny genetic scars that might be picked up just by the way you're doing sequencing. And it might be misdiagnosed as a point mutation based on some of these things as well. And so, so definitely, I think, a good point to make that, from the standpoint of, hold on, as we're thinking about mutation rates, to plug into some of the modeling, to think about when did this virus really spill over and come into humans? Was that as we're trying to like really kind of pinpoint that, was that in November of 2019? Was it in October? Was it in December? To really triangulate, to have a better measure makes sense. But I think, this, like, the conversation needs to continue and to grow with including even more of the biological complexity here, which is exactly what you're pointing out. And I think, reminds me of TWiEVO episode 2, when we were kind of dragged through the coals and it's true that there's so much going on here with the mutation rate, and it seems so simple to measure a rate, just as we think about like if there's a rainstorm outside and it's an inch per hour, that just seems like fairly straightforward. But mutation rates are much more slippery, much more difficult, especially as you kind of dig beneath the layers here.
Yeah. The word mutation rate for me means something different than it does in this paper, for sure. Because when I hear mutation rate, I go way down to the lowest level, and they don't. But
and then agreed. And then even kind of on that level, as you're pointing out, like then there's also it's like the old saying, like when a forest falls in the tree, when a mutation rate is measured in the absence of the host. So there are all these proteins that we deploy, the apobecs, which actually go in and are monkeying around with the literally causing mutations and patterns of mutations in a lot of cases. And there's some evidence of that for SARS-2 as well. And so the authors do point to that and at least partially account for that in some respect. But you can keep kind of adding layers of biology, and it just gets more and more complicated as you go.
Maybe it should be the error frequency when you're measuring polymerase, right? Just say, okay, how often does the polymerase put the wrong base in? And then, which would involve, I guess you could do the assay in the absence of the correction component as well, and you could see the really base error frequency, and then you put the correction protein back in, and you can measure that as well. And then maybe mutational frequency is when you get into the real world, right? When you actually make a virus.
No, but I love that study, like to pull back to, and I think that's possible. I think if the mutational basis of the proofreader is known, and then to run those proofreading and no proofreading, and then you could kind of work back and think about like how much proofreading is there really going on here? And like how important is that, right? Here, and this is coming up in the chat, asking exactly that question. We're on the same page.
So this has been done. People have measured polymerase error rates. I don't, and you're right. Not all errors are equally probable, but it depends on the neighboring bases. I don't recall the numbers offhand, but that's been done. Yes, for sure.
Oh yeah, of course. And then I think, that becomes interesting to me if you can make something like an apples to apples comparison between different virus types, or even like different kind of microbial forms, like to start to have because otherwise, what we see currently is a lot of times you just sort of folks go and sample the literature out there where a mutation rate is measured, probably sometimes in the population, sometimes at mutation accumulation. You can run this experimentally in the lab. So there's all these different from kind of distilled out, as you're describing, all the way to with all of life's complexity involved, very much an apples to oranges comparison. I guess my only other kind of, don't want to hammer on it too much, but note of caution is that, it's easy to measure point mutations, harder to measure the more sort of, complicated mutations, the recombination events, the structural variation, the indels, and how do you kind of collapse all those together, or do you hold them apart? That's a whole nother sort of, part of the equation, of course, especially with these proofreaders.
They also make an interesting observation about nonsense mutations, Nels.
Oh, yeah, that's right. Exactly. So, yeah. And that's kind of this funny thing, right? It's sort of the elephant in the room. So a nonsense mutation, right, is the stop, or would introduce a stop codon into a coding region, and sort of, depending on where that is, you might encode, the protein might go from, 300 amino acids long down to 5 amino acids. And that's probably gonna be a problem if this is a important protein. And so then how do you square the fact that, nonsense mutations, those are going to be purified out of the population, and yet the Ka to Ks or DNDS ratio is roughly 1. I think there, I guess the nonsense mutations you'd put in that category of weak selection, rare enough that that where they would be deleterious, that when you're taking the calculation across the entire genome, that might start to kind of go a little bit into the background. But it's also worth, and actually in our second half of the show, we're going to flip-flop from purifying selection potentially to a beneficial or a positive selection scenario with SARS-2. And so, and now we're talking about the spike gene, where a lot of, or at least there's some pretty conspicuous or obvious scenarios of receptor binding and spike processing and some related biology here for host recognition and virus cell entry.
Now, this mu variant has a stop codon in one of the non-structural proteins, and I just, I don't. What could you do with that? And so they say, nonsense mutations account for 10% of the missing mutations. What could you do with a few incidences here and there? These are obviously in proteins that aren't essential. So you can put a stop codon in and either get rid of them entirely or truncate them. So could you learn anything about frequencies from that?
Oh, good question. I'm going to punt on the frequency. So the frequency, I think but it's a great point, because the biology matters again, right? And it raises an important issue here, which is probably if you have a nonsense mutation, certainly if it's in something essential, it would very easily fall into that. It's deleterious to the viruses that encode that, and it's purified away. But you can also, and we've talked about this actually, there's not just SARS-2, there are known cases where if you have a nonsense mutation that can be beneficial to the viruses, can outcompete other viruses, because and so this is like super common actually in vaccine development for I'm thinking about pox viruses, one of my favorites. But so if you start to passage these things outside of their full host immediately, and these are massively large genomes, DNA based genomes, immediately you start to select for these deletions, stop nonsense mutations and deletions, where you gain some advantages by letting go of some of the genes and coding proteins that they're not essential, but even losing them is not just kind of neutral, it can be beneficial in some cases. And that's true for some coronaviruses, isn't it? What's the open reading frame that's commonly lost?
ORF8 is one of them.
Right. That's the one I'm thinking of.
But mu has a different one. I think it's NSP3, but I don't recall. I mean, that's supposedly an interferon antagonist, so you don't actually need it, and it could attenuate virulence in some ways, but who knows? The other thing that's
good.
The other thing, mentioning spike, they actually use one of the stories we did here on TWiEVO with Jesse Bloom, where they just trashed every amino acid in the receptor binding domain, remember? And they said,
Yes.
which can you touch and which you can't? And so they used that data to say, yeah, the number of changes we're missing is consistent from that study is consistent with what we're predicting. I think they predicted 8% versus 10% or something like that, right?
Yeah, exactly. And that's a really nice part of this paper. So where they do pull in some of the biology. And so this is, I think you're drawing back on, I looked this up ahead of time, TWiEVO 57,
Yeah, yeah.
where we had as guests Jesse Bloom from the Fred Hutch, and then Allie Greeley and Tyler Starr, postdoc and grad student. I think I flipped it. Tyler's a postdoc, Allie's a grad student, who did this incredible work, right? Where they put in like this deep mutational scanning. They put in all the mutations like along the spike, test them for binding, and ask, is there any, sort of improvement or loss? And, and that's right here in figure 3, as they're looking at the mutations specific to spike and making that point. So again, like, and it's worth it to, if you haven't heard that episode, to hear it from the mouths of the researchers. But so it's that kind of balancing act between some level of purifying selection. The spike, it needs to look like a spike in order to recognize ACE2. But at the same time, this isn't, evolution hasn't perfected the spike as it relates to binding the human ACE2. And so there they pulled out that N501Y, which increases binding. And then this is where I think, our kind of observations from the wild, from at least 3 different variants, I think alpha, gamma, and I'm forgetting the third, where you independently see that N501 mutate to Y mutation,
Delta
which
also,
has
yeah.
correct. Yep, exactly. That has, have been proposed for being part of the evolutionary process as the pandemic has been unfolding.
Nels, they have an aspect of this analysis involves what they call intra-host variation, where they actually go to the raw reads, right? Mm-hmm. Can you explain what they're doing there?
I think I might have missed that part of it. Did you?
So they
help me out?
The final sequence uploaded to
yep.
whatever database is what you've put together from the individual reads. And so they went back and looked at individual reads from each patient so they could see the intra-host variation.
Oh, sorry. Yeah, I'm with you now. Exactly. So that's right. So, and that gets to another layer of the onion here in a sense, right? So any one person, it isn't going to, you don't just pull out one coronavirus sequence. Viruses mutate, viruses change. You might even be co-infected, which has been highlighted from some of the data that's been gathered as well. And so, yeah, exactly. And so even within, it's almost like the single individual is kind of running an experiment themselves in real time. And you can start to look at the frequencies of the mutation. So, and that's where you would look as they're proposing that, for every 10 mutations that you see, 5 of those will be lost. Those will just be sort of blinking on in kind of that signal to noise zone. At really low levels. It's sort of like, how do you uncover something that's hiding or find something that's missing, basically. And so then when you're genotyping the virus, in most cases you're ending up, if you just want from one patient one final sequence, you go with the one that's at the height, all of the SNPs or polymorph, they're at the highest frequency, the consensus sequence across there. But there again, this is sort of a compromise for the level of information that you can actually wring out of this. And so, yeah, that was one of those kind of complementary approaches that they take to try to get a little bit of evidence in support of the, or evidence, I should say, that they found that does support their contention that for every 10 you see, there's probably 5 under the hood.
And it makes sense, because if you just look at viruses that are transmitted, you're going to miss all the variation that goes away when it goes to a new host. So you look within the host, and the key here is that the sequencing technologies allow us to do that, right?
Yep.
Which is beautiful.
It really is. I mean, it's incredible the amount of detail that you can get into. And that's, ultimately, I think there's some important discoveries there. We've started to see some. So that notion that there could be co-infection, that was, I think, unearthed because ORF8 was either in a single patient was either lost, there was a nonsense mutation or a deletion. But there were other ones where it was still there. And and then you could do that calculation. And then actually, that's how you could type recombination as well, is like to see to, especially if you're doing long read sequencing, where now you can say not only in a single read, not only is a mutation sort of hitchhiking along with the loss of orphate, you can really start to phase the genomes, as it's called, and really understand how these mutations can even travel together.
A couple of other things they hit on. One was that the changes seem, in terms of amino acids, that seem to be preferred. One of the conclusions is that it seems to be changing to match the human codon library.
Correct. Yeah, this is interesting. Yeah, and there's a kind of a corollary of this, which is this idea. So not all amino acids are biologically created equal. And so in addition to really obvious things like the amino acids you have at the spike interface with ACE2 will dictate the binding to some degree, there's also just properties inherent to the amino acids which will influence, how much energy does it take to put that R group, the thing that makes the one amino acid in the alphabet of, 21, 23, 21, I think, amino acids.
Yeah.
What makes them what makes each one different from each other, right, is the side chains. And those are molecules. And putting those together can have different metabolic costs, even to the host. And so that's something that they kind of took a peek at and commented on, is like, is there any hint of selection that you would like economize as a virus? So like it's almost this like crazy idea that you would, you don't want to be too rude to your host. You want to replicate like crazy and get out of there. You don't want to draw too much energy down from them. And so one sort of phenotype for that could be that the amino acids are less metabolically costly over time. That, I think they say there is like a little hint of that in the data that they analyzed, but it wasn't quite significant. And then your point too, though, that they do think that they start to see, so, each host has people will build these codon tables. And this actually is really important for when you're doing generating some of these estimates for mutation rates, is to have a sense of, so like when you're at the fourfold degenerate, even though you could imagine like that you'd have 25% of one codon for the amino acid, 25% of the other. That's actually not how it works out in the end. Those numbers aren't even. They aren't. And so then those are the codon usage. And that differs, for some reasons, are still, mysterious between even closely related species. And so, so you could, that could even be like a cryptic way to try to think about the spillover. Is there some, like, if you look at the codon usage of early days SARS-2, does that match the codon usage of a bat species? Whereas now it's moving closer to human. There's some problems with that, but
they didn't mention bats at all or any other species. They just said drifting towards humans,
correct,
which is interesting. And then the other is, as you've mentioned, the drift towards more stable, metabolically more stable amino acids, which they say, as you said, it's very small, but they say, well, if a virus needs to reproduce quickly, this could have some impact. But this virus doesn't reproduce all that quickly. It takes a cycle, I think, is 24 hours. So there's some phages that do it in 20 minutes. So I don't know how that works out, frankly.
Yeah, agreed. And especially when you're carrying along this 30 kb genome, that's like for an RNA virus, that's massive. And to get that all together, to get it proofread all together, packaged up and on to the next, that's like a high wire act. And so, it takes some time.
So Alex wants to know why we don't talk about quasi-species, but that's what we're talking about. What the enzyme does to make all the mutations in the RNA, that's a quasi-species, although the modern molecular biologists like Nels don't use that word anymore. That was my era.
Well, and then it was kind of rebirthed as an idea of like and this might be what Alex is getting at. So like the variation with, like, let's go back to the intra-host diversity, and that variation even within a single individual, a host individual, does that virus variation matter? So like, it's basically, as if you have this sort of cloud is kind of complicated. I think the reason we don't talk about it these days is it gets complicated fast, and it's kind of what seems intuitive can get a little bit counterintuitive. But basically, this collection of mutations, some on some genomes, some on others, and when you have that ensemble together, there's a fitness advantage relative if you were just a more homogeneous, genetically homogeneous population as the viruses. And so, yeah, I don't think the yeah, I don't. So, and this has definitely been put forward, and certainly for RNA viruses that have high point mutation rates, and are going crazy. I think I haven't seen much in that space for SARS-2, but I'd be curious. I'm sure people are kind of poking on that, but I think the amount of diversity we're talking about, even though it's very appreciable, and co-infections are possible, and there's even variation arising, of course, within an individual. I don't know how the quasi-species might fold into this, but yeah, interesting question for sure.
So a lot of people, Nels, are confused by in the chat by a lot of the things, and I can understand because you and I tend to just lapse into our way of talking. But maybe we could end this one with answering the question: why does it matter to know?
That's right.
Mutation frequency.
Yeah, it can seem pretty arcane, and definitely a lot of rambling or sort of trying to unpack it. And so, I think, and they put this forward, and I agree with them. So mutation rates matter because it's then an important number that you'll put into models or you'll put into other kind of equations to try to learn something more about the population dynamics. And so the example I think I already mentioned once or twice would be, based on the diversification of viruses that we sample today in real time, the collection, the sort of, if you just pull two marbles out of the bag or two genomes out of the GISAID database, and when they were sequenced, and how many mutations are different between them, and then if you also have a calculation of the mutation rate, you can start to work backwards to say, okay, given this many differences today, how much time does it take? Because it's mutations per site, and when you have those date stamps, then you add that, you can start to work backwards and say, who's the sort of last common ancestor of these viruses that we're sampling today, and when did it emerge? And that has obviously become, I think, probably one of the hot button issues with SARS-2, as people are debating, where did this come from, and how did it get here? Knowing the timing of that is part of the puzzle here. And so, and then I think, so that's a pretty obvious one. There's other sort of practical reasons for the mutation rate. I think it's I think it's important along the lines we've been talking about, which is as you now compare this to other types of viruses, other infectious microbes, other types of microbial life, to start to just get a handle on what are the sort of similarities or differences, and how do you, so all of, whatever virus you pick, if has humans as a potential host, all those viruses have to get around or replicate despite the same kind of immune defenses, and yet the mutation rates turn out to be pretty different, and the types of mutations that are accruing are pretty different. And so I think there's some real information in there about how viruses evolve and work, which is kind of fundamental knowledge hiding under here as well.
I also think that the technology, that the approach they use to find the missing mutations, right, is novel, and it can be used elsewhere. So I think
absolutely.
Sometimes you may not have other outcomes, general outcomes, but the methodology, the approach may be, and I think this could be a case where this will be refined. As they say in the discussion, this is probably an underestimate of missing mutations, but it'll be refined and used in other systems too. So I think that's also important.
Yeah, I agree, especially for the kind of real-time analysis, right, where the existing tools fall a little short. All right, so now we're going to flip the script here, Vincent, and we're going to go from purifying selection as sort of the accounting for the missing mutations to a potential case of positive selection, like a beneficial mutation that could have a big impact, and start to get at the $1,000,000 question that everyone's been thinking about and talking about: what's going on with the Delta variant, and do some of the mutations, in particular in spike, have some impact on virus spread? So the title of this preprint, which I think is in its third version, I'm guessing it's also making its way through peer review. This came out I think kind of late summer. This was updated September 5th, so it's still very actively being pursued. So this is delta spike. This is mutation proline, or P, at position 681 to arginine, or R. That mutation and spike enhances SARS-CoV-2 fitness over the alpha variant. And so first, let's just run to kind of highlight some of the authors here. So Yang Liu and Jianying Liu are co-first authors, senior author, co-senior authors, Zuping Zi and Piyang Shi. And they are all at the University of Texas Medical Branch down in Galveston, Texas. I also can't help but to lift up one of my good science buddies. This is Vineet Manachari, who's been at Galveston for a few years now. He was a postdoc in Ralph Barrick's lab, and one of the really great, thoughtful virologists, kind of evolutionary virologists who I think just does really great, rigorous work. He's been working really hard since the start of this pandemic and really, really going after some of these questions, and I think casting some light on some of these questions that we're all thinking about.
Here's also Scott Weaver, who's at Galveston, a well-known arbo-virologist who was on TWiEVO. We did a TWiEVO in Galveston just towards the end of 2019, and he was on it.
Oh, wow, nice. Yeah, we've been in touch. So Scott helped orchestrate a shipment of some virus samples our way, not SARS-2 related, but some other kind of run-of-the-mill common cold coronaviruses. And so, yeah, very generous, very kind, really good scientists. And I would say Galveston emerging as like a real leader in some of the virology here for SARS-2. They have all the BSL-3 facilities in place, a lot of the expertise, great scientists kind of putting this together. So and I think this is a really interesting example of what you can do when you put together some of this skill and talent and curiosity. And so, of course, the big question that we're all wondering about is, are there certain mutations or is the variation that we see in the Delta variant responsible in some way biologically for the increased spread? And of course, this has also become, you don't have to go too far into a TWiEVO podcast to start to get into some of the debate, whether or talking to Daniel Griffin as well about, and some of the clinical reports, thinking about, how do we weigh the really clear and pretty dramatic changes in human behavior. I think as right after the vaccines were unrolled and early in summer, when we kind of maybe collectively in a lot of communities sort of let down our guard in some sense, how much has human behavior influenced the spread of Delta versus actually the biology of the virus or the mutations, the variation there.
And as we have said here many times on TWiEVO, Nels, we've talked about changes in these viruses that give them greater fitness and therefore displace previous variants, as opposed to changes that facilitate spread through a population. And this is among some of the first papers to actually address that. We did one on TWiEVO, I think 2 weeks ago, where they looked at reproduction in cells and culture of Delta versus Alpha, for example. And they, that paper, which was in Nature, was nice because they actually did infectivity assays in human respiratory cell lines, but what's lacking in that paper is they didn't do competition, which we'll see in this paper is really important.
That's right. Yes, exactly.
So they did competition here, but they didn't do infectivity.
I know. It's really tough. There's so many moving pieces here. And so that's what I think we're, kind of rooting from the sidelines, trying to pull up the different research and see how it might kind of inform the other. But it's really true. It's really hard, that like killer experiment that kind of really gets to the heart of that question, to pull that off. Really difficult. So yeah, we'll kind of tackle it in this direction. And I would just, for our listeners who haven't, to check out that episode of TWiEVO, all of the others that have, there's all these thought-provoking discussions, really important as we're kind of, not only is the virus, evolving in real time, our understanding and our of it is a real-time kind of analysis as well. And so, and it's a really important conversation to keep having, especially as there are disagreements or assumptions and so forth, because we're not talking about as you're saying, Vince, it's not just one field or one set of experiments, right? It's all these
Yeah.
drawing from different fields and, the drawbacks and strengths of different experiments to try to put together a really complicated puzzle. So here, as you're saying, the authors, and really, in order to make some progress, I think narrowing down. So they really focus on it's about 10 mutations or so that distinguish the Delta variant from the Alpha variant in the spike coding region. So of course, there's many other mutations. I think we always love to sort of pause and remind ourselves that all of science has sort of put their eyeballs on the spike, but there's other really interesting biology. There's other important differences outside of the spike when you think about how a virus replicates and operates that are kind of, I think, underappreciated. But anyway, so very good, motivation to look at these 10 mutations. They actually make cDNA infectious clones where they start to kind of plug and play or mix and match, pull each of those 10 mutations. They'll revert them, from it'll be in the delta backbone of the whole genome, and then they'll put each of those 10 mutations that revert it back closer to the alpha variant at each of those positions, and vice versa. So to take the alpha variant and instead of having those 10 substitutions that would be consistent with what you observe in delta, just one of each of the 10 in the alpha backbone. And so and then they, as you're saying, so this is cell assays. So it's measuring the amount of RNA in competition. So it's not infectivity, and that's a very important caveat. And I think both points are, and we'll circle back to that in a few minutes. But to describe what they're doing here, so they basically have made those viruses, and then they do these head-to-head competitions where and here it is. So if we look in. Yeah, exactly. So that's what I was kind of describing, this mix and match process. And here we're just looking at, of those 10, they end up really zeroing in on this proline 681 to arginine. And so this is what the viruses look like. And the first two at the top is just sort of a more gross swap of the entire. So this is like all 10 of those mutations are now swapped into the delta backbone. They've reverted delta back to the alpha overall coding region of spike. And then you see the same process, but for delta, where they've reverted the arginine substitution back to the proline, the original P that you see in the alpha variant there. And then they start doing these assays. And so the competition assays, where you have relative replication fitness, meaning that they've infected, they know the starting input of their infectious clones for if you mix the alpha compared to the alpha spike and the delta backbone, or all of the different combinations, color-coded, aqua and red. And then you can ask after the infection runs for 1 day, 2 day, 3 day, 4 day, 5 day, and that's what you see in panels B, C, D, all of these different combinations of the head-to-head infections, roughly 1-to-1. You see that so if it was an even competition, that horizontal line at 1 is where you would expect the RNA values to come out, meaning that for every one RNA molecule of delta, there would be one RNA molecule of alpha, right. Et cetera. And so what you see is, as indicated in panel B, for example, just delta versus alpha. You can see that you put delta and alpha together, you recover more delta RNA. And then with that as a basis, you can start to play around, right, and start to mix these things up. And so, and what they narrow in on, and they're not showing all the data here, but that single position at 681, if you take away that arginine, make it a proline, now it does worse compared to delta, and you kind of do all these combinations of the backbone. And so
this one here, this delta, they revert it to the P, right? And that doesn't do well.
Is that?
I think delta P81 versus alpha, it loses, see?
Correct. Yeah, there it is in panel F there. It starts to go below that line. And so as the infections go on, so that's their evidence. And that was, for that one, it was the most striking, or the one out of the 10 that had that effect. There's also probably some epistasis going on here, meaning that, so the other mutations kind of buffer the ones that you have. So like you take it out of context and the thing does worse. In a couple of different ways. But anyway, so that's kind of the result, kind of a simple study. And then,
the thing is, Nels, that's a beautiful experiment
Agree.
if they had only measured infectivity.
Infectivity.
And
Yep.
why do I say that? Because you don't know what RNA copy level means, especially when you're making different viruses. You don't know how maybe one makes more RNA as a consequence of it. You need to measure infectious virus. I don't know why. And I bet if we asked the authors, they would make some excuse. Oh, we know it represents it, but it doesn't in an experimental setting. And I hate to
No,
harp
no,
on
I
this
hear you.
over and over, but
Yep.
it's really, otherwise it would be such a beautiful experiment. Go ahead now. Sorry.
Yeah. No, no. I hear you. I mean, it gets worse. So, um, this word fitness, which again, like is really important and has different definitions in different fields as well. And so this, again, we're kind of right back into why we're having these conversations, the TWiEVO space. So like in addition to measuring RNA, I agree. You'd love to see infectivity. There could be some peer review happening behind the
Yep.
scenes here
Yep.
that might be pushing it that way. But kudos to them for getting this information out right onto the preprint
Sure. Sure.
fast and updating it. Really an interesting finding. But then, so fitness really from an evolutionary standpoint would kind of almost necessarily put back all of the biological complexity. And so even if they did an infectivity assay, then we could still say, well, yeah, you did that in a human epithelial cell, or you did that in a adenocarcinoma cell line from a human lung. And so it's, obviously it's a great choice for a cell line, but the fit, like, in the real life fitness of how that single mutation might work out when, and not just in a cell where it's sort of like this viruses show up at the same time and it's sort of a homogeneous set of cells that they're infecting for a few days. What about if that was in a real human lung, or what does that mean for the $1 million question of transmission or spread? What does this mean for human to human spread? And so of course you can't do those experiments. You can't engineer a virus like this, set it loose into a human population, and then try to do this these awful kind of unethical courses. And so so it's sort of these trade-offs or this reductionist approach, and what is the information that comes out of here. So, and that's where I think I give them credit for going to this other step, which is to then tell us a little bit about, or to chase a little bit of the biology here. So so given some of the data we've already been talking about, like the mutation N501Y convergently has shown up, or independently has shown up in a few different of the variants, and has been measured by Jesse Bloom's lab and others for increased binding of the spike to ACE2. That's sort of a attractive hypothesis. But what they're saying here is that actually no, for proline 681R, the binding to ACE2 is the same. But there's a different property that they propose which matters here, and that is that position 681 turns out to be in the furin cleavage site. And if you measure the cleavage of which in that site divides the spike protein into two segments, S1 and S2. And if you measure how the efficiency of that cleavage, if you have an arginine versus a proline at that exact position, with the arginine, it's a more efficient cleavage of the furin of the furin site. And so that's actually a different phenotype, or a different sort of functional outcome from some of the other mutations that have been described so far and proposed to be functionally consequential.
Yeah, I like that we're starting to get more experimental science here and just getting away from the epidemiology and the numbers driving all of our interpretations about the biology of the viruses, which isn't correct.
Yeah,
so now
right.
Over the next couple of years, we're going to see more and more of this, and it's going to start to make sense. We're not there yet by far. It's going to take a long time before any of this gets into the textbook. But yeah, this change appears to affect the efficiency of furin cleavage, which they speculate allows more efficient fusion of
Correct.
the viral and host cell membrane. So that makes sense, and that could impart a fitness advantage, right? For sure.
Yeah. Yeah, and Vineet, so not just this preprint, but the Vineet's lab has really been pursuing the furin cleavage site and really dissecting that, not just at this position, but others. And I think doing some really important work in that direction as well. But yeah, it's still, we're in sort of, we're in virus real-time evolution here. If you think about scientific time, where we're still less than 2 years into this, I mean, science moves slowly. And I think we can both point to many virological studies in our careers that took more than 2 years to get to the bottom of something. And so it's sort of this mismatch in the things we can observe in real time versus like to really kind of glean an understanding and put together some of the functionally consequential, probably the product potentially of positive selection here, where you're selecting on something in the population. Why is it that the virus is displaced? Why did Delta displace Alpha and, the variant game that we've been playing?
Some of the, I mean, that's so true, Nels, that it takes time, and that runs up in a pandemic. That runs up against the desire to get answers overnight, right? And you can't do it, and that's part of what's been driving the press, right, and all the the international stories. So it makes me cringe because you don't have answers, and
I know. Yeah.
I mean, the one answer we do have is we have vaccines, and they work.
Yeah,
and,
that's right. No, exactly right. And I think that's kind of, you're getting at a little bit of the heart of some of the friction here, is that mismatch in time, and that's led to, I think, a lot of misunderstandings. And so, in some sense, I think, the lessons of this pandemic are what we learn from SARS-2, which we didn't, we didn't do this in SARS-1. SARS-1 didn't have nearly the impact. MERS even kind of less so. And also the difference in technology then. But so I think maybe, I mean, one way of putting it, it's sort of, maybe disappointing or kicking the can down the road, but it's like, what we learned from this SARS-2 pandemic, from a virological standpoint, from the virus evolution standpoint, will probably be most useful for SARS-3 or SARS-4 or whatever pandemic might be done. And this is kind of how basic science works, how fundamental science works. It's the stuff we learned, 10 years ago, 20 years ago, was massively important for like launching this incredibly successful mRNA vaccine, from design to FDA approval. And like that turnaround, I think we almost take it for granted. It's just still catches my breath how, like, just what an incredible kind of moon landing level, once in a generation almost, scientific advance there on the shoulders of work that was 20 years ago. And I think this kind of stuff that we're talking about today will probably have that echo into the future, of where the real value of it isn't for that kind of ripped from the news headline. It's from, how do we deal with the stuff that's ahead that we don't even know about yet.
Yeah, and also the source, I mean, not just the science, right, that allowed this to happen so quickly, but the sourcing of materials that you need to make all this, right? To make billions of doses of vaccine, you need to get the raw materials. And there are people who specialize in that, and that's become an art in itself, right?
Yeah.
Product pipeline people and so forth. So it all came together. For sure. I want to just acknowledge a couple of people who have supported us today here on TWiEVO. From JL, nothing like a live stream TWiEVO to help wind down after the last day of the European Society for Medical Oncology annual conference. Thanks and all the best from Paris. Thank you for your contribution.
Oh, beautiful. Thank you.
Any of you would love to support our work? We do these science episodes on our own time. We do it so you can hear science conversations and perhaps learn a bit. And I'm of course continuing to do it in a new space called the Incubator. So your contributions are really going towards keeping that going forward, because we're not the press. We don't get advertising support. We do it on our own. So we need your support. And if you like what we do, help us. Sandy, thank you so much for your contribution. Wow. Those that can do science, those that can't support TWiEVO. Love it.
I love it. Thank
There's
you.
There's a little button
Sandy.
at the lower, just below the video window. There's a little button, actually below the chat window. There's a little button with a $sign. You can click that and donate anything you want.
No, it's wonderful. I would just, speaking about us doing it on our own time, I was in a really fascinating first in-person seminar of the season here in Salt Lake in Utah, and kind of kept my eye on the clock and jumped out a few minutes early so we could jump on here and podcast together.
Oh, thank you. Appreciate it.
Oh, it's my pleasure. I couldn't wait to get on. And so this is really fun.
So Bill makes an interesting point. Do you think it's interesting that Alpha with P681H and Delta with P681R have been the two most fitness dominant variants so far? So first of all, Bill, I want to thank you for using fitness, any other word. because I think that's what you can conclude. And you know, it's interesting. 681R is an improvement on 681H, according to these results, right?
Yeah. Yep. No, I think that's I would point to more of Vineet Menacherry's work, um, talking about the furin cleavage site, tongue twister there.
Yeah.
And that's, that kind of got, we talked about this a while ago, right? This got your old mentor into some hot water a ways back. David Baltimore kind of made a comment about the furin cleavage site, but if you do a deeper dive on this, there's some, and I think we're still learning so that efficiency that is shown in this paper as you're stepping through from proline to either histidine, the H, or in case of the delta, the arginine here, what that means for the efficiency of the cleavage and how that then the processing of this to go from the spike fused to S1 and S2 that then are part of that recognition of ACE2 in that process. I think we're still learning what the real outcome is.
Sure.
And so, but it's incredible to think down to that single amino acid resolution that this really is starting to crop up when you see, and that's from the evolutionary standpoint, right? When you see or different, in this case, it's not the same mutation, but it's at the same position.
Right.
And that you see that can have an outcome that sort of starts to, I think, promote that in its potential importance functionally.
Remember this change arose randomly, right? Somewhere. And it most likely has been positively selected because it conveys some kind of fitness advantage. And we're going to see maybe a change somewhere else outcompetes it. Who knows? This is all happening and we're looking at it. So it's going to be interesting.
Yep.
By the way, Nels, we now have 99,000 YouTube subscriptions, and that's cool because that 100,000 is kind of a cool number, and we've been ripping through the higher 90s in the last couple of weeks. So thanks, and thanks for joining us. And this is a good point. Most people are better at auditory learning than reading. That was kind of the idea behind starting all these pods, that it's a lot easier to listen to someone. And in fact, even when there's no visuals, it's because, you go to a meeting and you look at slides, you tend to not listen to the person talking or vice versa. But I think if you just listen to people chatting, it can help. At least I find I learn well. So
I do too. A couple of other, side effects of that as well. I think I mentioned this when you had me on a guest a while back on TWiEVO that, I think Rich Connett's voice saved my life. So I was driving when you're driving somewhere in the Mountain West where I live. I think I was on the road to Montana to give a seminar at the University of Montana. Hours of driving. I probably was starting to nod off at the wheel a little bit, and I was playing podcasts, and somehow on my old iPhone setting, all of a sudden TWiEVO just came up, and it was Rich's booming baritone voice that sort of got me to kind of startled me awake, and probably kept me from swerving off the road there. So
that's good.
Yeah, there's some practical side
on the other hand, we had a letter recently, Nels, to TWiEVO where someone said they use us to go to sleep at night. So
well, that's great too. So I like this idea of these long. Speaking of long form, we're now north of an hour here, but you can kind of think about these podcasts or these conversations as almost. I kind of think about it as the little fireplace in the corner of the room that is just, there's this kind of warmth emanating from the conversation, and you don't have to pay attention to it for the whole hour plus, but you might just for a few minutes say, oh, that sounds like an interesting detail, and kind of your attention might come up, or you might rewind 15 or 20 seconds or whatever, but then kind of let it stream. And just, even if it's providing a little bit of company, to me, like I get a lot from podcasts, just from even that kind of, just that companionship almost through the earphones, and so, or through the stereo. So I think, there's a lot of really fun sort of side effects here of podcasting, actually.
Thank you. How would you say it, Auden or Odin? Do you have any idea, Nels?
I think closer to Odin. I'm terrible, as you know, on name pronunciations, and even though my own heritage, so we're looking at some Norwegian Kroner here. Even though my own family can trace back to both Sweden and Norway, my language skills are along these lines are pretty terrible. So I'm going to just say a big toximicket. Thank you very much.
Nels, you're hitting the mic there.
Oh, sorry. Yeah, you're right. I'm
Sorry.
getting excited. We've been on this podcasting hiatus, our summer break, and so I'm kind of bubbling
Dig
with
Down has
excitement.
been listening since March. Thank you, Dig Down. By the way, if you don't want to give money, that's okay, but you can
Yeah.
give us a thumbs up. There's a little thumb thingy at the bottom of the video window. We have 157 people here. We could get over 100. That would be great. That helps us. And yes, TWiEV and TWiEVO are food for the brain. You're absolutely right. And we're glad that you like it. We're just
Yeah,
chatting and trying to be clear.
good to see you here again, Rima. Yeah.
You want to do some picks, Nels?
Let's do that. So, my science pick of the week is this. It's a symposium, but it's more than that. It's called the Leading Edge Symposium. It's this group of a growing group of postdocs and other scientists. So, the Leading Edge is an initiative to improve the gender diversity of life sciences faculty in the U.S. by providing women and non-binary postdocs with a platform to present their work, an opportunity to meet and connect with one another, and mentorship and career development training for world leaders in biomedical research. And so the reason it's a great group and the reason I'm lifting this up is so I had the honor of being part of a panel earlier this summer with the Leading Edge Fellows as part of their symposium and an incredible discussion. It's one of those kind of experiences where, as you know, as we all inevitably get older, as we age through our careers, you like come across young people that just inspire you, that they're just so ahead of the curve, and you kind of you're like you can kind of surrender or let go in some sense. So whatever tiny bit of career advice just from sort of being around a little longer I was able to give, I think it was repaid back tenfold, and just sort of the inspiration and the energy that this group is bringing to their science, and and starting to address some ongoing and really difficult imbalances. So, anyway, this is my pick of the week, the Leading Edge Symposium for rising stars in science. Yeah. How about you, Vincent? What's your pick of the week?
So my pick is based on last Friday's TWiEV, where we had two modelers talk about vaccine equity. You know, how to the effects. They modeled the effects of two countries sharing. One country, you know, has a lot of vaccine. The other country has none. And what would happen if the country with gave more and more vaccine to the country without? And it really makes an impact on the infection rate. And this was Shadi Saad Roy and Carolyn Wagner. We had both of those on the show, and you can see on this paper a lot of well-known authors: Michael Minna and Eddie Holmes, Jeremy Farrar, and so forth. But this is the app that you can use to put your own numbers in and see what happens.
Wow!
So it's an interactive scenario here. You know, you choose your parameters, and then you get different modeling, right? And it uses their framework, and you can go put your own numbers in, right? So you would go here to the dashboard, interactive plots, and then you could say how many doses, the period between dose, the week, and so forth, and then you click do it, and you get evolutionary scenarios, and you can vary the parameters. Very cool, I think.
Yeah, this is fantastic.
And anybody can go to this.
Yeah. Wow. Yeah, what a great interactive way to illustrate how, I mean, it's kind of intuitive, right? That sharing vaccines for across the world for a pandemic that is traveling the world makes sense. But yeah, what a great way to dig in on that and to really see the potential impact here. That's a great one. Yeah.
We also have a listener pick. We have a pick from Thomas Hoffler from Free University of Berlin. And this is, I'm going to pull up the Amazon page here, The Gene's Eye of Evolution by J. Arvid Agren. Now, the A with the little circle on top. How would you say that?
Right, that's right. Again,
I'm not asking the right person. Right?
You know, if you find me, like several times I've gone to Stockholm in Sweden, which is an incredible, really fun city, and surroundings. You know, if let's say I'm just kind of hanging out in someone comes up to me in public and mistakes me for a Swedish person who might speak the language, I just have like a flight or fight response where I just run away, because I should just say I don't speak the language, but I just sort of panic like this. But so Arvid, yes, this is, thank you, Thomas, for lifting this up as a guest pick. So Arvid's been in touch with me, and Vincent and I are working now to host him as a guest on TWiEVO this fall. So stay tuned. If I don't think it's going to be our next month's episode, but before the end of the year or right around the new year, I think we will. I can't wait actually to welcome Arvid, who I've met and been at a meeting or two together. Really a big fan of his work, and haven't yet had a chance to read Gene's Eye View yet, but it's on my priority list here, and I'm already hearing really positive early returns, great reviews, and I think we're gonna have a blast covering this and welcoming Arvid.
Krister points out you can model hesitancy too in that modeling site that I just showed you.
Yeah, vaccine hesitancy,
cool.
Which now kind of underlines our, the case in the U.S. What a mismatch in the abundance of resources versus the uptake of them. Yeah.
John says, looking forward to your lecture. So I'm going to live stream virology here on YouTube, and
oh, sweet.
I promised to start in September, and I think I'm going to be able to make that. We're almost at the end. We have a little more than a week left, right? And so probably we'll start the beginning of next week. September goes through next week. So
you've still got time.
I got time. I'm going to do it live stream here on YouTube. It's going to be from the incubator, and I'm going to do my virology lectures, but you're going to be able to ask questions. We'll have quizzes. We will have exams. Yes, you're going to go online and take an exam if you'd like to test your knowledge. Good. And it will be on my YouTube channel, and it'll start next week. I'm probably going to do it at 11 a.m. Eastern time to accommodate both the West Coast and Europe. I'm sorry, Asia. I don't know how to get to you.
Yeah, that's tough. Yeah, might have to do.
But of course, they'll be recorded, and you can review the chat and so forth. But I'm looking forward to that. It's an experiment to see if you can, teach virology in a live stream format.
I think it's great.
I mean, I'm going to show slides as I usually do, and then we'll take breaks for questions, and I'm sure there'll be a lot of questions. And it'll be recorded and kept there, and so you'll be able to go back and see it later. So the slides will be provided. Should be fun. It's an experiment. As I said, if you guys say, we don't like this, change this, that, we can change it midstream. It's no problem.
Well, you've been piloting this experiment, with this live streams, both with Amy and TWiEVO and your other podcasts, and it's already clear, even in a few months here, just sort of, it's almost like adding a whole nother gear to the mix to see the stream of comments coming in, to kind of curate and put those up. That's a skill. It's already.
It's going to be twice a week. It's probably going to be Mondays and Wednesdays. 2 hours roughly I'll go for. My lectures are usually about an hour, so I figure with the chat we're going to go to 2 hours because you're going to ask a lot of questions. And I would like to have the questions related to the virus, right? So, if we're talking about what is a virus, I don't think you should ask questions about should I get 2 doses or 3 doses of vaccines. Hold that for the vaccine lecture. Right?
Yeah, that's right. And this will be arranged virus class by virus class. Is that the organization?
No, it's going to be the first course, which just principles of virology. It's going to be by principles, right? We're going to go through the reproduction cycle. Then we'll go through how viruses cause disease, vaccines, antivirals, evolution, emergence. We'll have a lecture on therapeutic viruses, ecology of viruses. And then if it works, then the next class I would teach about specific viruses, a more advanced course. And I would even do a course on emergence. Let's look at all the viruses that have emerged over the years and see what the principles are. So I plan to do this as long as I am able. Right?
Yeah, we've got 160 or so here. Big recommend. I know I'm going to be tuning in. This sounds like a lot of fun.
It would be 11 a.m. Eastern time, right?
Mm-hmm.
Mondays and Wednesday. I will put up a marker on YouTube soon by the end of this week. And yeah, I need good moderators. Yeah. Are they oriented towards lay people or pros?
Mm-hmm.
Well, I think a lot of people, I have my Columbia virology lectures are up on YouTube already, and you can get a sense from what they're oriented at. But, the fact that it's a live stream can make it easier. So you say, I don't understand at all what you're talking about. And just ask, and I'll explain it. It's going to be a bit different from a lecture, so I can take more time and do that. So,
Well, and I think it's a great question, and part of the experiment, right? So I think not only will you be providing some really interesting information, these lectures, this teaching experience, but we'll also be learning from it. How do we communicate the concepts of virology, and how does, to a layperson or all kinds of different backgrounds, what is it that connects, what's confusing, and that back and forth, that conversation, which we don't do enough of, could really, I think, break things open a little bit in terms of how we, as scientists, as virologists, as evolutionists, et cetera, can do better at reaching more people. Yeah. What a great experiment.
If it works, I would, in the future, bring guest lectures in, right? Because get people who are experts in certain viruses to talk about what they're doing. That would be, the advanced course. So a lot of possibilities. And someone asked here, is there a web page? I don't have it up, but I should get it up by the weekend so you can see what we're. But if you want to go to my blog, virology.ws/course, that's where all the archives are previous virology courses, and you could check those out. Are any pre-lecture readings required or recommended? No, you could just come and start absorbing it up. I think that would be fine. Or you could check out some of the lectures already, but I wouldn't. I'd just come in and bring your questions. That's all.
Yeah.
And, I'm sure we'll have Nels in the future. Next course, Nels will give a lecture on using DNDS to sort out virus-host interactions.
That's right.
Nels?
Count me in. As you know, I'm here and ready to go anytime.
So that is it for this TWiEVO. You can find the show notes at microbe.tv/TWiEVO. If you want to send a question or a comment, those are kind of dried up since we do the live stream now. People just come here.
Which is great.
TWiEVO at microbe.tv is where you'd send an email or any other questions. And as I said, if you'd like to help us, if you can use the chat, the what is it called? The super chat or
chat
super stickers. Button. Or if you don't want to do that, you can go over to microbe.tv/contribute. We have a couple of ways you can help support us. And again, this goes towards supporting what we do. That's all. And Microbe TV, by the way, is becoming a nonprofit. Sometime this year when the IRS gets its act together, and then your contributions will be tax deductible, and we won't have to pay any taxes also. Of course, I couldn't do this without Nels Elde. You can find him at cellvolution.org, and over on Twitter, he is L Earlybird. Thanks so much, Nels.
Thank you, Vincent. Good summer break, but I can feel the wind in our sails. We're kind of going into the fall academic season here, so looking forward to the next one in a few weeks from now.
I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiEVO is by Trampled by Turtles. You can find them at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious. Bye, everybody.